Okay, hey everyone. As we continue through our Unit 2 video series, and on our last video, we talked about pedigrees and how pedigrees are used to trace some type of gene through a family. And they're used to predict the probability that certain individuals in the family will have that trait. And remember, traits are determined by our genes, which are segments of DNA. And pedigrees are typically used to trace a genetic disorder or disease through a family. And so we learned all the different specifics about pedigrees, but today we're going to do some actual case studies and look at some actual pedigrees of families that have genetic diseases that run through them. And we're going to see how a pedigree can be used to predict whether or not a child may be born with this particular disorder. Okay, so I'm going to share my my document camera screen with you. And we're going to look at this case study. And now, before I read about the case study, let's just review real quick what all the different parts of a pedigree are, because that's what we talked about in the last video. So just a real quick review. Each row of a pedigree is a generation. Okay, and so usually pedigrees have Roman numerals out to the side to tell you how many generations are shown in the pedigree. Circles are females, squares are males. If a circle and a square are connected by a horizontal line in the middle, like this, that is a couple, married or unmarried, but they are a couple that are able to have children. And so the vertical line that comes down from that couple indicates their children. So from that vertical line, you have this horizontal line that connects to their children. Okay, so they have 1, 2, 3 children, and all 3 of their children are married. You can see that here. All right, and then from them, you've got the grandchildren of the original couple, and then you have great grandchildren of the original couple. Okay, so we have 4 generations here. So circles, females, squares are males, and if their circle or their square is shaded, then that means they are affected by whatever that trait is. And if they're unshaded, they're not affected. So this is a pedigree showing people in a family that are affected with cystic fibrosis. So what is cystic fibrosis? Well, let's read this little passage so we can learn a little bit about it and find out what the pedigree is trying to solve. So cystic fibrosis is a progressive genetic disease that causes persistent lung infections and limits the ability to breathe over time. In people with CF, a defective gene causes a thick, sticky buildup of mucus in the lungs, pancreas, and other organs. In the lungs, the mucus clogs the airways and traps bacteria, leading to infections, extensive lung damage, and eventually respiratory failure. In the pancreas, the mucus prevents the release of digestive enzymes that allow the body to break down food and absorb vital nutrients. Cystic fibrosis is a genetic disease, so that means it's inherited from parents to children. People with CF have inherited 2 copies of the defective CF gene, 1 copy from each parent. Both parents must have at least 1 copy of the defective gene. Both parents must have at least 1 copy of the defective gene, making the gene for CF autosomal recessive. So in this passage, they tell us that CF is an autosomal recessive trait. Sometimes they don't tell you. And so that's when you have to go through those questions to figure out, and we'll review those questions as we work this pedigree as well. People with only one copy of the defective CF gene are called carriers. So one copy of the defective CF gene makes them a carrier. And remember from last time, a carrier is heterozygous, meaning they have one dominant allele and one recessive allele. So big A, little a. Since CF is recessive, then carriers do not have CF because their normal gene or their dominant gene compensates for the faulty one. Okay, so here we have a pedigree that illustrates how genes for diseases like cystic fibrosis can be passed from parents to their children. Okay, so if they had not told us that cystic fibrosis was autosomal recessive, we could have figured that out using our questions that we learned in the last video. So remember the question we need to ask is, are there children who are affected with neither parent affected? And so if I start down here at the bottom, I see that these two are affected, but neither of their parents are affected. And this one is affected, but neither parent is affected. So that tells me that it's recessive, but now I have to figure out, is it autosomal recessive or sex-linked? And so the next question I ask is it all males or like mostly males that are affected? And I see that there are 1, 2, 3 females affected and 2 males. So this one is not sex-linked or X-linked. It is on the regular chromosome, so it's autosomal recessive. Okay, so using the letters, it tells us using the letters big A and little a, what is the genotype of the following people? So we're just going to label the genotype of all the people, just like we did in the previous video. But we're going to start with, remember there are 3 possible genotypes. There is little a. There is big A, little a. Okay. And then there is big A, big A. But in a pedigree, it is very rare that you can determine absolutely positively the big A, big A. So in those cases, we use big A dash because we're not sure. We know that the individual has the dominant trait, so they have at least one big A, but we don't know for sure if they have big A, or if they have big A, little a. So in that case, we use that dash. Okay, so let's go through and figure this out. We know that cystic fibrosis is autosomal recessive, so that means those that are affected with cystic fibrosis have to have 2 copies of the recessive gene. So the ones that are affected have to be little a, little a. So I'm going to go through here and label the ones that have little a, little a. And that is the ones that are affected by cystic fibrosis. Now, all the other ones are not affected. So that means they have at least one dominant gene. So all of these other ones get 1 capital A. And we have to figure out what their, we have to figure out what their second letter is in just a minute. So, but I'm going to go through here and I'm just going to give all of the unshaded their capital A because they all have at least one dominant gene. Okay. So now I have to figure out which ones are the big A, little a. Okay. So which ones are the carriers? Remember, carriers are heterozygous. All right. So the way you determine if someone is a carrier is you look at the ones that are little a, little a. So you go around and you look at all the where you have little a's. And if an individual is a parent of a little a, or they are a child of a little a, then they have to be big A, little a. Okay. So if we look down here, we see this girl and this boy are little a. So that means they inherited a little a from each parent. So remember, you always get one copy of the gene from your male parent and one copy of the gene from your female parent. So if you have two recessive genes, then that means each parent has to have at least one recessive gene. So each parent is a carrier. Okay, so the same thing for this one. This one has two little a's, and so this is his parents, so they each have to have a little a. Now, if I come over here to this little a, this girl, she has two copies of the recessive gene. So each of her parents has to be a carrier and have a copy of the recessive gene. Okay, now if I look at this one has two copies of the recessive gene. So we know that one copy of the recessive gene came from his mother, but the other copy of the recessive gene had to come from father. So father is a carrier. And so now if we look at this one has a child that is not affected because this child has one big A, one dominant. But because this girl's mother has little a, she has to have a little a. Okay, and then now if we look at this one's parents, we go back up here and we look at the parents of this one, we see that since this one has two copies of the recessive gene, then each of her parents has to have a copy of the recessive gene. Okay, so now all of the rest of these are going to be A dash because we don't have enough information to determine if they're big A. Because if you come down here, you see that this girl and this boy have parents that are big A, little a, big A, little a. So they could have gotten a big A and a big A, or they could have gotten a big A and a little a. So this one and this one are a dash. Okay, and we don't know about this one's parents or this one's parents, and their children are big A, little a, or just big A. We don't know enough to say for certain what they are. So these are going to be dashes, and then all the rest of these are going to be dashes. So most of the time in a pedigree, you can eliminate this. The only time you're going to use the big A is if everybody in the pedigree is big A. That's the only time. So you can pretty much forget about this because that doesn't happen very often. And if it is happening, then there's no really reason to use a pedigree because you know what everybody's going to be. So most of the time you end up using the A dash for the ones that are not carriers. Okay, so now we can answer these questions. Well, no, we can't because we don't have person. Okay, so we've already pretty much answered this. And is cystic fibrosis dominant recessive? We already know it is recessive. Okay, so let's look at another case study now. Let's move on to a different pedigree. Okay, so this time, this is a pedigree for Huntington's disease. And before we read, let's just look at this pedigree and see if we can figure out if Huntington's disease is autosomal, dominant, recessive, or X-linked. Okay, so remember our first question is there anywhere where the there's a child that is affected and neither parent is affected. So I see here that this boy is affected, but the daddy is also affected. And then I see these two siblings are affected and their mom is affected. So the answer to that question is no. There's nowhere on there where a child is affected and their parents, neither of their parents are affected. So it's not recessive. And so therefore we know it's also not X-linked. We've got two females and two males equally distributed. So it is dominant, autosomal dominant. So now let's read the passage about Huntington's disease. Huntington's disease, or Huntington's chorea, is a rare autosomal dominant. So again, in this passage, they tell us that it's dominant, but we know how to determine it if they don't tell us. It results in the slow deterioration of the physical and mental abilities of the affected person. Experts estimate that 1 in every 10,000 people, about 30,000 Americans, suffer from Huntington disease, and another 150,000 are at risk for developing it. Many describe the symptoms of HD as having ALS, Parkinson's, and Alzheimer's simultaneously. So it's a pretty bad disease. Symptoms usually appear between the ages of 30 to 50 and worsen over a 10 to 25-year period. Ultimately, the weakened individual succumbs to pneumonia, heart failure, or other complications. Every person who inherits the HD gene will eventually develop the disease. That's because it's dominant. So you can't just be a carrier of it. It's a dominant trait. So even if you're heterozygous, you have it. Over time, HD affects the individual's ability to reason, walk, and speak. Since the disease does not manifest until a late age, an affected person will often have children already. So you can take genetic tests to see if you're at risk for the disease. So this is a really good example of how pedigrees can be used to be helpful for people. Because if you have a disease like this running in your family, you really would like to know if you could possibly have it, because you're not, a lot of times people don't develop symptoms until they're 50 years old. And by then you've probably had children. And so even if you have it, it's a really good chance that you're going to pass it on because it's dominant. Okay, so to work this pedigree, you have to do it opposite of the way you did the autosomal recessive pedigree. So that means that the people who are affected get the capital A this time because they have the dominant gene, because Huntington's disease is dominant. And so the people that are not affected get little a. They have two recessive genes, and so they don't have it because it is dominant. So if you don't have it, you have two recessive genes. So you notice how this one is exactly opposite of the one we did before. The little a's are going on the unshaded individuals, and the big A's are going on the shaded individuals. So now we look at all of the big A's and we try to figure out, okay, who are our carriers? Because even though carriers will have the Huntington's disease, there's a 50% chance that their children will not have it if they're a carrier, because they might give them that recessive gene that does not have the disease. Okay, so we ask the question, is the person connected to a little a, either as a parent or a child? Okay, so if we start right here with this one, we got to look at this one's children because we don't have their parents. So this one has 4 children, 1, 2, 3, and 4. And we see that 2 of this one's children have little a, and they don't have Huntington's. So that means that this one, although this one has Huntington's, this one has the recessive gene. So you see how for this one, this male does not have Huntington's. The mama does have Huntington's disease, but some of her children did not have it. 2 of her children did have it, but 2 of her children did not have it. So it's a 50-50 situation here. So that's why you would want to do a pedigree to see if you were heterozygous, because then you have a 50-50 chance of having a healthy child. Okay, now if we look at this one right here, although this one has Huntington's disease, this one's father does not have it. So that means this girl is a carrier. Okay. And if we look at this male has a father that has 2 little a's. So he is also a carrier. And then if we look at this one down here, this one has a mother that has the 2 recessive genes. So this one is also a carrier. So this person right here now can find out that they are a carrier. And so that means even though they're sick, they have a 50% chance of having a child that is not sick. Okay. So let's look at how we can determine that 50%. So go to this next question. Okay, so if person number 3, okay, so that's this person right here. If person number 3 has a child with someone who does not have Huntington's, complete a Punnett square to determine the chance that their offspring will have Huntington's. So, sorry, give me a second. Okay. So they're asking if person number 3 has a child with someone who does not have Huntington's. So person number 3 is little a. And so a person that does not have Huntington's, which genotype are they out of these 3 possibilities? Okay, so Huntington's is dominant. So both of these are going to have Huntington's disease, right? Because it's dominant. So this one will be healthy. They will not have Huntington's disease. So if person number 3 has children with a person who does not have Huntington's disease, then we can do a Punnett square. And we can see that all of their children will also be little a. So they have a 0% chance of having Huntington's disease. So even though this person has a dad that has Huntington's disease, if they marry someone, see, they don't have it. So if they marry someone that doesn't have it, then there's a 0% chance of Huntington's disease passing on through this person. Okay, even though their dad and their grandmother has it, they are not going to, if they marry someone that doesn't have it, their children are not going to have it. Okay, so that's good news. And that's how a pedigree can be used to help people. Okay, so now this last one, okay, before we read the passage, I want us to look at the pedigree and try to figure out if it is autosomal. I'm having a hard time here, guys. Hold on. Okay, here we go. So we're going to look at this pedigree and determine if it's autosomal recessive, autosomal dominant, or X-linked. And so we say, are there children who are affected and neither of their parents is affected? And the answer to that is yes. So we have a boy here and that is affected, neither of his parents is affected. A boy here that's affected, neither parent is affected, and a boy here that's affected and neither parent is affected. And then we ask the question, is it mostly or all males? And yes, we have only males are affected. All of the females are unaffected. So that tells you this is a sex-linked trait and it occurs on the X chromosome. Okay, so let's read about this trait. Hemophilia. Hemophilia A is a genetic disorder caused by a missing blood clotting protein. People with hemophilia A often bleed longer than other people. Bleeds can occur internally into joints and muscles or externally from minor cuts, dental procedures, or trauma. According to the U.S. Center for Disease Control and Prevention, hemophilia occurs in approximately 1 in 5,000 live births. There are about 20,000 people with hemophilia in the U.S. All races and ethnic groups are affected. The X and Y chromosomes are called sex chromosomes. The gene for hemophilia is carried on the X chromosome. Hemophilia is inherited on an X-linked recessive manner. So females inherit 2 X chromosomes, one from their mother and one from their father. Males inherit an X chromosome from their mother and a Y chromosome from their father. That means if a son inherits an X chromosome carrying hemophilia from his mother, he will have hemophilia. It also means that fathers cannot pass hemophilia on to their sons. So fathers give their sons a Y chromosome. So if a father has hemophilia, he will not give it to his sons. But because daughters have 2 X chromosomes, even if they inherit hemophilia from their mother, most likely they will inherit a healthy X chromosome from their father and they won't have hemophilia. So since hemophilia is recessive, if you have at least one dominant gene, the hemophilia gene is recessive. And so the dominant gene covers that up and you don't have hemophilia, even if you have that recessive gene, if you have the one. And so since girls have 2 X chromosomes, they have a good chance that one of their X chromosomes will have the dominant gene, even if the other one has that recessive. And so girls are not as likely to get hemophilia as boys for that reason. A daughter who inherits an X chromosome that contains the gene for hemophilia is called a carrier. So she can pass the hemophilia gene on to her children, but most likely she'll pass it to a boy rather than a girl. All right, so let's look at this pedigree and let's label the genotypes of the different individuals. So whenever we're doing sex-linked traits, we use X's and Y's along with the letters when we do the Punnett square, which we're going to do in just a minute. So, but for now, we're just going to use the letters. And so we know that hemophilia is sex-linked recessive. So that means the individuals that have it have we're going to give them little a, and I'll show you what to do with that in just a minute. So little a here. Okay. And so the ones that don't have it have at least one dominant gene. So they get the big A. Okay. Now we have to figure out who are the carriers or who's heterozygous. So if they are connected as a parent or a child of a, then they have to be a carrier. So if we look down here, this boy is a, so that means mom and dad are carriers. This boy is a, so mom and dad are carriers. This boy is a, so mom and dad are carriers. These we don't know. So these are all dashes. Okay. So let me show you how you use the X's and the Y's. Okay, so dad only has one X chromosome. Okay, so in this case, dad doesn't get two letters. So all the males get one X chromosome. So the males are XY. Okay, and the females are XX. So let's just go ahead and let's put XY over with all of our males, XX with all of our females. Okay. So with our females, we know that one of their X's has the dominant gene and the other X has the recessive gene. With males, we just need to figure out what's on that X gene because that Y doesn't factor in here at all. So if they are not affected, then that means their X has to have that dominant gene on it. So their genotype is X, capital A, Y. Instead of instead of A, big A, little a, they're X, big A, Y. The females are written like this, X, big A, X, little a. Okay. So we're just going to go through here and fix the males. So this male is not affected. So this male is X, big A, Y. This male is affected. So this male is X, little a, Y. This male is not affected. So this male is X, big A, Y. This male is not affected. So this male is X, big A, Y. This male is affected. So this male is X, little a, Y. This male is not affected. So this male is X, big A, Y. And this male is affected. So this male is X, little a, Y. Okay, so that means the genotype for person one is a female that is not affected. So there are two X's, but we also know that this female is a carrier. So this female is X, big A, X, little a. I know it gets confusing with the X's and the Y's. So person two right here is a male, and this male is affected. So this person is X, little a, Y. They don't have another X chromosome because they're a male. Okay, so let's do a Punnett square problem using this pedigree to predict the probability that a child will be born with hemophilia. Okay, just a little bit more. Okay, I know this looks like a bunch of gobbledygook, but we only need to look at one particular person. So if person number 3, okay, so person number 3 is right here. If person number 3 has a second child, a daughter, complete a Punnett square to determine the chance this child will have hemophilia. Okay, so person number 3 is a male that does not have hemophilia. So his genotype is X big A Y. Okay, that's a male that is not affected. Okay, and it says, what is the probability they will have a second child who is a girl? What is the probability that this child will have hemophilia? So his wife over here is X big A, X little a. His wife is a carrier. Okay, his wife doesn't have it, but she is a carrier of hemophilia. That's why they ended up with a son that had hemophilia. Okay, so these are the parents' genotypes. So let's put those over here on the Punnett square. So X big A, Y, X big A, X little a. So this is how you do a Punnett square if it's a sex-linked trait, because you have to consider whether it's a male or a female on these traits. So let's bring in our X big A, bring down another X big A, bring over X big A, bring down X little a. The X always goes first. So bring down X big A and bring over your Y. Bring down X little a, bring over your Y. Okay, so notice that 2 of your 4 boxes result in females and 2 of your 4 boxes result in males. It's a 50-50 chance, right? Every time you have a child, it's a 50-50 chance, male or female. Okay, so what will the genotype be of the female child is what we're looking at. So we're wanting to find out if they have a second child that is a daughter, what's the chance that the daughter will have hemophilia? Okay, so in this case right here, this daughter will not have hemophilia because this daughter inherited 2 dominant genes, and hemophilia is recessive. So this daughter will not have hemophilia. And this daughter also will not have hemophilia because even though this daughter got 1 recessive allele, they also got the dominant allele. And so the dominant covers up the recessive. So this daughter is not going to have hemophilia, even though she's a carrier. So the chances of them having a child, if it's a girl with hemophilia, is 0% for a girl to be affected. But look at if it's a boy. So if they have another child and it's a boy, if the boy inherits the dominant allele on the X chromosome, they're not going to have hemophilia. But if the boy inherits the recessive allele on that X chromosome, they are going to have it. So it's 1 out of 2 if it's a boy. So a 50% chance that a boy will be affected. Okay, so that's why with sex-linked traits, boys are more likely to be affected than girls. Okay, that's it for pedigrees. So, and that's also it for our Unit 2 genetics series of videos. So on the next video, we'll move into Unit 3, which is evolution. So thank you for your attention, and we'll see you next time.